This is Ritik, your biology subject matter expert from MEMONEET, and in this video, we will be discussing the remaining part of the cell cycle and cell division. Before getting into the remaining part, so we'll just recap what was the previously we have discussed. So previously we have gone through what a cell cycle and how our cell is made up of, and how exactly the living organisms are made up of, and what are the major important aspects and roles of the cell, right? Then we have gone through with the what is cell cycle and what are the phases that do comes with the cell cycle. Then we have also considered with the phases of cell cycle, right? Then what do you mean by M phase or mitosis phase? Then we have also gone with the G1 phase, S phase, which is nothing but the synthesis phase, then G2 phase, G0 phase, and lastly, we have also concentrated on the entire M space is nothing but the mitosis, right? So in this video, we will be concentrating on the next part, that is meiosis. So what do you mean by meiosis? The meiosis is nothing but the production of offspring by sexual reproduction, including the fusion of two gametes, right? So, few gametes instance, if we are concentrating about the human reproduction, so how the production of the offspring has been done? So, it is either by the fusion of sperm and the fusion of ovum, right? So, fusion of two gametes, each with a complete haploid set of chromosome. So where those gametes are formed from a specialized diploid cell. So this is a specialized kind of cell division that reduces the chromosome number. So here it is actually reducing the chromosome number. Say like if there is 46 chromosomes, so reduction division is happening. Wherever the reduction division is happening, it is subjected to be the meiosis. Okay, like 23 set of chromosome you will get. So here also, if you have 46 set of 46 chromosomes, so it will come like 23 chromosomes. So totally what is happening? The chromosome is getting reduced. This special kind of cell division that occurs or that reduces the chromosome number by half, resulting in the production of haploid daughter cell. This kind of division is actually called as meiosis. I hope you're clear what is meiosis. So meiosis ensures the production of the haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores what? Diploid phase. So now we have like say two types of sexes. Say like it could be the female and it could be the male. So once what happens, these two gametes they combine, right? Now whatever the reduction of this chromosome were there will be restored. So from 46, how much chromosome was that? 23. Once the fusion happens, again you are going to see 46 chromosomes. So we come across the meiosis during the gametogenesis in plants and animals. And this leads to the formation of what? Haploid gametes. What is the major key feature of the meiosis? So meiosis, it involves two sequential cycle. So one is the nuclear and cell division, which is called as the meiosis one, and the another one is meiosis two. But only a single cycle of DNA replication. That means, so even though the meiosis involves two sequential cycle, one is meiosis 1 and meiosis 2, but DNA replication happens only one time, that is single cycle of DNA replication. So meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatides at the S phase is nothing but the synthesis phase, where the meiosis involves pairing of the homologous chromosomes and recombination between non-sister chromatides of homologous chromosomes. That is meiosis 1. It initiates after the parental chromosomes have replicated to produce what? identical sister chromatides at the S phase, where this meiosis 2 involves pairing of these homologous chromosomes and recombination between these two sister chromatides, okay, of homologous chromosome. Then the four haploid cells are formed at the end of the meiosis 2. So meiotic or meiosis event can be ground under the following phases. So in meiosis 1, you will see prophase, metaphase, anaphase, telophase 1. Whereas in meiosis 2, again you are going to observe what? Prophase 2, metaphase 2, anaphase 2, and telophase 2. What is it? We'll see. See, in prophase 1, what happens? The prophase of the first meiotic division is typically long and it is more complex when compared to the other phases of the meiosis 1. So, it has been further divided into the five phases based on the chromosomal behavior. So, that is the very first thing you are going to observe is leptotin, zygotin, pachytin, tryptotin, and the last one you are going to observe is diakinesis. So, in general, what do you mean by leptotin? So, during leptotin stage, the chromosomes become gradually visible the way you are seeing here under the light microscope. So the compact of the chromosome continues throughout the leptotin. So here chromosomal condensation. So the thread-like chromosomes, they start getting condensed and the chromosomes will be visible under the light microscope. Then this leptotin is followed by the second stage that is called as of the prophase one is actually called as the zygotin. So what happens in zygotin? During the stage, the chromosomes, they start pairing together. And this process of association is called as synapses. Very important. What do you mean by synapses? The pairing of the chromosomes together is actually called as the synapses. Such paired chromosomes are actually called as homologous chromosomes. So pairing of such chromosomes are called as what? Homologous chromosomes. So in the zygotin, what happens? The chromosomes pairing and synapses begins. That is pairing of the chromosomes will, you are going to observe this. So electron micrograph of the stage indicates the chromosome synapses is accompanied by the formation of the complex structure called as this is the structure that you are going to see here, that is synaptomenal complex. Clear? Synaptomenal complex. So the complex formed by a pair of the synapsed homologous chromosome is called as bivalent or tetrad. However, they are more clear visible at the next stage, that is in the pachytin, you are going to observe the clear visibility of it. So during this stage, what happened? The four chromatides of each bivalent chromosomes become distinct and clearly they will be appears as the tetrad in position. So this stage is characterized by the appearance of recombinant nodules, the site at which the crossing over takes place. Like we have this chromosomes here. So this is a site where crossing over you are going to absorb like this. So this is actually the stage where you are observing what? The crossing over. Okay. So can you see here? In the pachytin stage, the site at which the crossing over occurs between non-sister chromatides. This is chromatide is different and this chromatide is different. Crossing over is happening, right? So that is what you are going to observe, the crossing over between two homologous chromosomes. Okay. So crossing over is also an enzyme mediated process, and the enzymes which actually involve is called as recombinase. So this is very important, a neat kind of question. Which is the enzymes which is involved in the mediated process of crossing over? That is recombinase. So crossing over leads to the recombination of the genetic material. That is nothing but exchange of the genetic material between the two chromosomes. and the recombination between the homologous chromosome is completed at the end of the Packeton stage, you are going to see that recombination is completed, and now the leaving the chromosomes linked at the site of crossing over. Next we have is after the Packeton stage, the beginning of the diplotin, that is, this stage has been actually recognized by the dissolution of the uh, synaptonymal complex. What is a synaptonymal complex? Is that the tendency of the recombinant homologous chromosomes of the bivalent to separate from each other. That means now what happens? Two homologous chromosomes, they have crossed over. Now they have formed recombination. Now they have to get separated, right? So that thing you are going to observe in the diplotin. So the X-shaped structure are called as chiasma. So whatever the structure that you are seeing here is actually called as chiasma. X kind of shape is called as chiasma. So what happens is in oocyte of some vertebrates, the diplotin can last for more than a month or an year. The final stage of the meiotic prophase 1 is actually the diakinesis. So what happens here? So this diakinesis is actually marked by the terminalization of the chiasma, that is separation of the chiasma. During this phase, the chromosomes are fully condensed and the meiotic spindles is assembled to prepare the homologous chromosomes for the separation. So now in this process, you will see the complete separation of this homologous chromosomes which are been attached. By the end of the diakinesis, what happens? The nucleus disappear and the nuclear membrane, whatever you're seeing, right, envelope also gets breakdown, and the diakinesis represents transition to the metaphase. Okay. So after prophase 1, you are going to observe the anaphase 1. So the homologous chromosomes, what happens here? They get separated while the sister chromatides remains associated at their centromere. Then we have telophase 1. In telophase 1, what happens? The nuclear membrane and the nucleus reappear. Then the cytokinesis follows, and this is called as dyad of the cell. Although in many cases, the chromosomes do undergo some, kind of dispersion, they do not reach the extremely extent state of the interphase nucleus. So the stage between the two meiotic division is actually called as interkinase and is generally short-lived, short period of time. So there is no replication of the DNA here during the interkinase. So it is followed by the after prophase, anaphase, telophase, right? It is followed by prophase 2. Much simple when compared to the prophase 1. So we have prophase 2 here, which is the meiosis 2. So what happens in prophase 2, as you can observe here? So one thing that you are going to observe here is two kind of division will occur. So meiosis 2 is initiated immediately after the cytokinesis, usually before the chromosome have fully elongated. So in contrast to the meiosis 1, after meiosis 1, the meiosis 2 will occur. So which is somewhat normally like the meiosis 1 itself, but the nuclear membrane disappear by the end of the prophase 2. The chromosome gains again become the compact. So coming to the metaphase 2, what happens? At the stage, the chromosome align at the equatorial. So can you observe here? Same like meiosis 1 itself, it is going to same. So the chromosomes align at the equatorial and the microtubules that you are seeing, right? So they move opposite poles of the spindle fibers. They get attached to the kinetochore of the sister chromatides. So this is the kinetochore of sister chromatides. Then we have anaphase 2. In anaphase 2, what happens? This is anaphase 2. We are observing anaphase 2 here. So it begins with the splitting of this. So this, if this is the centromere, so they get split. So splitting of the centromere of each chromosome, which was being holded by what? Sister chromatides together, allowing them to move towards the opposite pole. So they will start moving towards what? Opposite pole, right? Of the cell by shortening of the microtubule attached to the kinetochore. So the last one you are going to see is telophase 2. This is the telophase 2. So in the end of the telophase 2, in which the two group of the chromosomes, group 1 and group 2 chromosomes, once again get enclosed by the nuclear envelope. Again, you are going to see the nuclear envelope surrounded. Then the cytokinase form, resulting in the formation of tetrad of the cell, that is four haploid daughter cell. So how many are there totally? Four haploid daughter cell you are absorbing. Then what exactly is the significance of the meiosis? So what is the major significance of the meiosis here? So meiosis is the mechanism by which the conservation of the specific chromosome number of each species is achieved across the generation in especially sexually reproducing organisms. Even through the process, okay, they're paradoxically resulting in reduction of the chromosome number by the half, but it is the one, this meiosis is the one which is actually helping to restore, to store the what do you say that the genetic character. So it also involves in the genetic variability in the population of an organism from one generation to the another or next generation. Then the variations are very important for the process of evolution. So meiosis only are going to observe the process of what? Variation. So this was all regarding the what do you say that meiosis, how exactly the cell, what is the major role of our cell. So what is the importance of cell division, what is mitosis, and what is meiosis, and what is yes phase. So this was all about the cell cycle and its regulation. So if you feel any doubt, you can comment to the comment section, message box, or else you can also reach me out through the Telegram channel. Thank you.